Okay, we are going to look at Chapter 3, Movement of Substances Across a Plasma Membrane. So still under the basics of biology. So this Chapter 3 has got 4 subtopics, and we will be looking at the first one, and that is the structure of the plasma membrane. Okay, first, this is the cell, this is the cytoplasms, and you have nucleus, and you get to see arrows over here, arrows going in and out. So the question is that why do substances need to go in and out of a cell? Okay, so you can see the answers right here. It says many biochemical reactions takes place in a cell. And because of that, they require many substances to carry out this biochemical reaction. Okay, and because of the reactions also, there are some waste products that are being formed or produced, and those waste products cannot stay long in the cell. They must be excreted or eliminated out. Otherwise, they are going to be toxic to the body. And therefore, these are the reasons why substances need to move across the plasma membrane in and out of the cell. Okay, so the first one, to make it simple, is to obtain nutrients and gases needed to carry out those biochemical reactions, to excrete or to eliminate metabolic waste, as well as to maintain a suitable pH and ionic concentration, or in other words, to provide them with an optimal condition for the cell to carry out its activity. Okay, so again, this is a cell. We have inside the cell over here the cytosol, the cytoplasm, and also the surroundings of the cell, which are the extracellular fluid. So you can see arrows in and out, and these are why substances, or this explains the necessity of the movement of substances across a plasma membrane. Okay, you see water uptake or water loss, secretion, respiration, nutrition, ions, as well as excretion. So these again summarize the necessity of movement of substances across a plasma membrane. Now let's look at the structure of a plasma membrane. Okay, this is the cytoplasm, the inner of the cell. Can you imagine that? And this is the environment just now. They are the extracellular environment. Extra, that means outside of the cell. And this is the structure of the plasma membrane. So what are the main components that form a plasma membrane? Okay, as you can see right here, the blue thing. Okay, these are actually the lipid component or the fats component. You call it the phospholipid. The phospholipid. Also, you have this brown color. Okay, this structure that are the proteins. So that means main component of a plasma membrane are the lipids, the phospholipids, and also proteins. Okay, this is outside of the cell and this is inside of the cell. Can you imagine that? Now let's another picture to show the structure of a plasma membrane. So mainly phospholipids, and you have here embedded embedded in between these phospholipids are those proteins. Okay, let's look at a phospholipid molecule. Okay, there's a head and also the two tails over here. The head likes water, feel water. Hydro refers to water. So that means the head likes water. So it is attracted to water. The tails, phobic, phobia, dislikes water. Okay, again, go back to the structure. And you get to see how are they arranged to each other. The head, hydrophilic, the tail, hydrophobic. And they are arranged in this manner, double layer. So it is double layer of phospholipid, or in other words, phospholipid bilayer. So you get to see the tails here facing one another, and then the head. The environment is fluid, but this one is the inner side of the cell, that is the cytoplasm. Okay, another head over here which likes water just now. They are facing the extracellular or the extracellular fluid. So the tails here is in facing one another. They dislikes water. Okay, phospholipid bilayer. A polar head, non-polar tail. Okay, according to the fluid mosaic model, the protein molecules float within the phospholipid bilayer, forming a mosaic pattern that changes frequently. So in other words, when you look at this seems like rigid and also solid, but then actually they are dynamically moving, or in other words, we say that they are actually flexible, changes frequently. So this is called the fluid mosaic model. Okay, also we have two types of proteins over here. The channel protein, okay, this one, usually you see this the channel over here. Okay, let's see if we have other picture. Protein that shows to differentiate that. Okay, the channel protein has got a tunnel, a channel that allows certain substances to go in and out. Also, you have the carrier protein. Usually, schematic diagram is we draw it in this manner with a specific structure or shape to bind with certain substance. So in other words, the structure of the plasma membrane, mainly formed by the lipids and also the proteins, they are changing frequently, constantly. Okay, phospholipid layer. Then this is called bi, that means two layer, outside and inner of the cell. This is the one. They are not rigid or static, but they are dynamic, flexible structure with protein molecules float about it in a phospholipid bilayer, forming a mosaic pattern. So that means they are always changing like fluid. So therefore, they are called the fluid mosaic model by these two scientists that discover the plasma membrane. Okay, that is on it for 3.2. Okay, let's see. Sorry, 3.1 here. Movement of substances across the plasma membrane. So you get to see another structure that shows the plasma membrane, where you see the phospholipid arranged in a bilayer manner, and then you get to see those proteins embedded within. You get these are the molecules, small molecules and ions, non-polar molecules like oxygen, carbon dioxide. Then you have water over here, lipid soluble molecule, the larger molecules. So here different types of substances are shown, and they are telling you an information or a message is that this plasma membrane is generally described as selectively permeable. Okay, what does that mean? It only allows certain substance to go through it because of the nature itself. Just now we say the head likes water, hydrophilic. Okay, so what if the substance is too big? Can it pass through or diffuse through just by simple diffusion, just diffuse in through this phospholipid structure? Okay, how about those ions? How about those with charges? So you have to understand that because of the structure of the plasma membrane itself, okay, it doesn't allow all substance to pass through it. In other words, they are selective. Separa telak ini, selective, or certain substances sahaja yang boleh pass through it. Okay, for fat-soluble molecules and small molecules, such as oxygen, they are able to pass through unaided. Okay, apa maksudnya? Unaided tu tanpa bantuan, just diffuse in like that. Okay, again, look at the substance. What kind of substance? Fat-soluble molecules. Because we know that these phospholipids is actually fats, right? So they can just diffuse through those fat-soluble molecules. Just diffuse through. And if they are small, they have no problem sisip masuk. Begitu sahaja. Okay, water molecules can also diffuse across the plasma membrane. Water molecules, kenapa? Okay, water molecules because the head yang kepala itu, the phospholipid head, just now we say they are hydrophilic in nature. Okay, another one. Okay, water-soluble substances such as glucose and amino acids and ions need to be aided by carrier protein. Okay, although glucose and amino acids, they are water-soluble, but they don't just go through the phospholipid bilayer just like that. They have to be aided by carrier protein. Why? Questions will ask you why. Then your reasoning is that these substances are too big to diffuse through. Okay, kalau ions, they also need to go through special pathway like carrier protein. Okay, so let's look further into that. The channel protein, the carrier protein. So which of these substances can pass through phospholipid layer or carrier protein? What do you think? Ions. We have the water, we have those glucose together with amino acid just now through this carrier protein. How about those common salts? So you ask whether they are water soluble or fat soluble. Okay, carbon dioxide, maybe they are small enough, just diffuse through the phospholipid bilayer. How about vitamin E? Fat solubles. Okay, so if it is fat soluble, just diffuse across. How about glucose? Just now already mentioned glycerol, fat solubles. So you have to start and to begin classifying these substances and to identify which pathway are they going through. Okay, there's this experiment that you will be doing, okay, to study the movement of substance across a selectively permeable membrane, and we will be using this whisking tube, which has this permeable nature. And this is the setup of a simple osmometer, and we are going to observe the level of this sucrose solution, the rise or the fall in the level. Okay, describe further on that. Now before that, okay, let's go back to see the structure again. Okay, common factors that determine whether a molecule can pass through a plasma membrane is by their size, by the polar nature, whether they are polar or non-polar. Okay, for example, water molecules, they are polar. Non-polar molecules, such as oxygen and carbon dioxide, but because they are small, okay, by the size, so they can just pass through the phospholipid bilayer. Okay, how about if they are large, such as glucose and also amino acid? Okay, so they cannot just diffuse across the diffuse across the phospholipid bilayer. They have to be aided by the carrier protein. Okay, how about the non-polar molecules such as fatty acids, glycerols, those fat-soluble vitamins or steroid compounds? Since they are fat-soluble, so they can just diffuse across the phospholipid bilayer. How about those ions? Okay. Okay, we go to see this. There is also this cholesterol structure. Okay, now what is the function of cholesterol? Okay, how about this glycoprotein? Since this is protein and this is the 6 carbon, this is actually depicting carbohydrates. So that's why here glycoprotein. So when this carbohydrate structure is attached to the protein, so that's why we call it as the glycoprotein. Okay, see, yeah, the cholesterol structure is here. It is to make sure that the structure of this plasma membrane is not too fluid, because just now we say they are not static, they are not rigid, but that they are flexible and has this fluidity structure. But because of the presence of cholesterol, this will prevent the plasma membrane to be too fluid, and hence it stabilizes the structure of the plasma membrane. Okay, so that's it about the structure of a plasma membrane.